FMN, FES, and N2 are all inside the enzyme, whereas Q is a carrier. It's a molecule that is free to move around the membrane. It's not stuck in the protein. So essentially, the process of complex 1 is to move it from NADH to Q. So that's the first thing that complex 1 does. The other thing it does that we don't see here directly is that it pumps 4 protons to the intermembrane space. The exact mechanism for how it does the proton pumping is not relevant for the class, but just note that it pumps protons, so it fuels the gradient. So the electrons in this step of the reaction lose energy. That energy is used to pump protons. What about complex 2? So complex 2 is a little special. Complex 2 doesn't actually use the product of complex 1. You can think of the two of them kind of functioning like this, where you have complex 1, then complex 2, and then instead they both merge into complex 3. So it's not necessarily linear in that sense. That's why I wanted to talk about them together. So complex 2 helps us use FAD instead. So if complex 1 is an NADH replenisher, complex 2 is the opposite. So the way it works is the electrons from FADH2 are given to Q. Just like the electrons from NADH are given to Q, we are giving the electrons from FADH to Q as well. But here's what's really cool. We talked about the succinate dehydrogenase reaction and how that this molecule has an FAD on it. Surprisingly enough, this enzyme of the TCA cycle is actually attached to complex 2. So basically, as succinate gets reacted, the electrons directly go to FAD. And then from FAD, it goes to the iron sulfur clusters. And then after it goes to the iron sulfur clusters, it ends up to Q, to become QH2. So the final product is that both complex 1 and complex 2 produce mobile electron carrier QH2. I put in here a summary of what are the electron transfers that are present. So for example, complex 1, NADH to FMN to FES to Q. Whereas for this one, because it's attached to the TCA cycle enzyme already, it's actually direct, succinate to FAD to FES to Q. And the one thing that's unique about complex 2, no protons are pumped. And the way I like to remember it is because FADH2 is a very weak electron carrier. It doesn't have the same power as NADH. And so as a result, it is no surprise that it's not capable of pumping protons. So this is the origin of the fact that NADH is known to produce more ATP than FAD. And it's this is a manifestation of that. But where do we leave off from here? Our electrons have now been removed from NAD and FADH2, and now they're in QH2, ubiquinol. So where does QH2 go from there? QH2 then runs through complex 3 and 4. And here I will summarize with you the basic pathway. It's kind of just moving electrons to a different carrier, so I would just highly recommend that you put it on your cheat sheet. So complex 3 is known as cytochrome C reductase. It's not surprising that it has the reactant name or the product name in here. So basically, we take Q, so actually it should be QH2. We take QH2, we transfer the electrons to what's called the Q cycle. Basically, it's a set of electron carriers inside complex 3. Van der Heyden usually doesn't have time to go over it, but feel free to read it on your textbook. And then afterwards, once the electrons are in the Q cycle, they pass it on to cytochrome B, and then that is passed on to cytochrome C. So essentially, the net reaction is electrons are moved from QH2 to cytochrome C. And so because cytochrome C gets reduced, hence the name cytochrome C reductase. This process pumps 4 protons. The way I would understand this complex is through this electron pathway and the protons it pumped. Complex 4 is actually the last one. The only reason this is super important is that this is where we produce water. So cytochrome C passes its electrons to copper. Copper passes it to cytochrome A. Cytochrome A passes it to B. B passes to iron copper cluster. And then the last acceptor is oxygen. The one thing that we should note here, QH2 carries 2 electrons. Cytochrome C carries 1 electron. So in this scenario, in this pathway I've written here, 2 electrons are passed at each time. But in cytochrome C, to this step, only 1 electron is passed at each time. So in this diagram, Q is a 2 electron carrier. Cytochrome C is a 1 electron carrier. That's one thing to remember. The other thing to remember is that not only does this complex pump, but it produces the reaction we want. It puts the electrons to oxygen to form water. So this is part of the reason why we make water. Well, we digest our TCA and glycolysis pathways is because the last step of the electron transport chain. So to repeat, what do you need to know? Complex 3, you need to know that it's QH2 to C, cytochrome C basically. It pumps protons, and we've transferred it from a 2-electron carrier to a 1-electron carrier. Then complex 4, we transfer it from cytochrome C to oxygen to form water. It pumps protons, and it's the end of the electron transport chain as we know it. But all of that work we've done so far is to pump protons from the matrix to the intermembrane space. So let's talk about now, what do we do with that gradient? And so complex 5 is where it comes in. It's the ATP synthase. So essentially, we have a ATP generating mechanism where we have a rotor and a kind of a channel for protons to pump. And so the way I like to think of that is if I were to draw my membrane and my ATP synthase is written like this, if I were to divide this into 2 parts, the so-called F0 is a channel. It's where the H plus travels through. And then this part right here is the rotor. It's what spins. And so what the goal of the ATP synthase reaction is to use the power of the channel that the channel receives from the flow of H plus, you can think of it like water flowing, and use that energy to spin the rotor. And this is very, very applicable to think of as a hydroelectric power plant. It does the same pathway, except instead of water, it's H pluses that flow. So let's talk about the channel first. How does the channel let the protons flow down? But also at the same time, how does the channel produce a spin that we want? And so the way to think of that is that the channel has 2 parts, a kind of like spinning part, a rotating series of aspartic acid helices that are shown here as cylinders that are blue, and then a small section in the side. And this small section has 2 half channels. One channel is open to the cytoplasm, but not the other side. One side is open to the matrix, but not the other side. And so if we think of aspartic acid, this has both a protonated form and a non-protonated form. So how do we take advantage of this to create a flow of protons? So aspartic acid in normal pH is negative. It's deprotonated. So hopefully you guys remember that. So the way this works then is when it's positioned like this, because this intermembrane space is high in protons, the aspartate on that like half channel that is exposed to the high proton concentration will get protonated, as seen here. The H plus moves in and protonates that. So ta-da, you now have an H there. Great. Then as we move to the next side of the column, when this H moves to the next side and this H over here moves to the next side, we are confronted with this scenario. Again, from this point of view, this deprotonated aspartate is facing the high H plus concentration on the outside. So it's not surprising then that H plus will come in and protonate that. On the other hand, the H on this channel has a proton on it, but it's exposed to the low concentration of H plus on the bottom. So it's no surprise for us then that the H, or the proton, will leave this aspartate residue and become deprotonated. And so the net result is that we have a new proton come in from the channel over here and an old proton leave. And so the net effect is that we pull from the outside and we release on the other side. If you do not completely understand my English and how I describe this, I would highly recommend reading the textbook, as it gives a pretty clear definition as well. But essentially, if I were to track how an H plus would move in this channel, the way I would see it is an H plus would come in on the half channel facing the intermembrane space. Then it would spin around until eventually it reaches the half channel facing the matrix, and then it comes down over there. And in order to have the H plus move down, it must move in a circle. And this movement and mechanism is what generates the spin. This is the reason why the flow creates a spin. It's because the aspartates are not allowed to release it to the matrix directly. They must spin around to the other half channel in order to allow it to leave. So what did we answer? We answered, how is H plus taken from the intermembrane space? We answered, how is H plus released to matrix? And then we answered, how is spin generated? So now, what we're left off with is a spinning energy. Yeah, it's a rotational energy. How do we use this rotational energy to make ATP? Well, now you don't look at the channel, you look at the rotor. So let's look at this section now. So essentially, the rotor is structured in a way where there's a gamma protein in the middle, and it's a ring of alphas and betas. So 3 betas and 3 alphas. Okay, and essentially the way it works is there are 3 faces present in this particular section. The beta usually doesn't have anything. Instead, it's the alphas that are involved. So in one alpha section, we have ADP and P come in. And then this is often called the O form or open form. This alpha is in the open form because the gamma subunit has the configuration to turn it into an open form configuration. So essentially the presence of 3 alpha subunits that are the same in sequence but different in conformational change is possible because the gamma subunit is asymmetrical. And so this face of the gamma subunit makes this alpha subunit an open form, meaning ADP and P can freely come in. The other face of the gamma subunit is what's so-called locked. So in the locked form, it already has ADP and P, but it's locked, as in it cannot freely leave. So basically, once the ADP and P come in, they're kind of locked in place in there. The other last end of the gamma subunit is what's called the T, or like called tight or tense. Essentially, it is this part that is believed that the alpha subunit is so tight that it forces the ADP and P to come together and form ATP. So the first thing I want to describe is that this ATP rotor has 3 configurations in the alpha channel: an open form, a loose form, and a tense form. And it's all dependent on the gamma subunit and how it faces them to push them towards that configuration. So how does this make ATP? So let's imagine that we are an ADP and ATP on this configuration. So let's clear my drawings real quick. And let's just say that we are an open ADP. So initially, the ADP and P will enter the O state. Then the gamma subunit will rotate, and now we're in the locked phase. So now the ADP and P that just entered are now stuck in place in there. Okay. Then the gamma subunit is going to rotate and rotate again until eventually this L phase becomes a T. And so that is present in this part right here. Once that gamma has rotated to the point that it has conformationally changed this into a T state, it will look like this reaction. The ADP and P becomes ATP. Then once we're in the T state, it will rotate again to open to the O state. And once we're in the O state and you have ATP, the ATP leaves. And so if this is not clear, let me just show you again in a diagram form. So first we go from open state, then we go to loose, then we go to tight, then we go to open again. So what happens at each step? So first step, open, ADP and P enter. Loose state, ADP and P are trapped. Tight state, ADP and P converted to ATP. And then lastly, open state, again, ATP released, and ADP and P come in again. So this is how the cycle goes. And this is only possible, this change in pathway of going from O to L to E to T is only possible by the fact that the gamma subunit is spinning. And how is the gamma subunit spinning? Because it is directly linked to the channel. And that makes the spin of the channel translate to the spin in the gamma subunit. If this was not clear, I highly advise to watch a video. But what's most important is that this is an explanation on how mechanical work driven by the movement of the protons is used to form ATP. We're going to talk about shuttles at next recitation. I'm going to start off with this in a review. So hopefully we can cover that later on. But if you have any questions or any confusions on my recitations, feel free to email me. My email is very accessible. I have not really received any emails recently, but feel free to email me if there's any confusion in my recitation or if there's any improvement that you think you can I can work on. But yeah, thank you so much for your time, and I'll see you guys next week.